Good morning, dear students. Hope you will enjoying your good health. Well, student, in our previous lesson, we have introduced and we have discussed about the biological problem. Today, we will study an example of the solving a biological problem. As you all know, students, that the steps involved in solving a biological problem, that was the observation, hypothesis, and deduction, and last step was the experiment. So student, an example of a biological problem solving, that is malaria. Students, the term malaria, it is derived from two Greek words, mala. Mala mean bad, era mean air. So it was a belief that this disease, it is caused by the bad air about 2000 year earlier. It was believed that this disease, it is caused about 2000 year earlier in the bad airs. So some of the observation related to this disease in those ancient time. By the ancient time mean, as I already told, that it is about 2000 year earlier. There were the first observation about the disease, that was the chill and fever. Second observation about the disease was this was occur in the people who live in the low marshy places. Later on, students, some people voluntarily drank the water of the marshes, but they did not got suffered with the malaria. So on that basis, another observation established, which state that the stagnant water of the marshes, that what the stagnant water of the marshes poison the air, and people get suffered from malaria. So students, these all the three observations, that was about 2000 year earlier. Now, with the advancement of the biological sciences and the discovery of the different tools, nowadays means in about 200 year earlier, about 200 year earlier, about 200 year earlier, it was found that most of the disease that was caused by most of the diseases are caused by the microbes, yeah, or bacteria. So a French scientist whose name was Laurent King, A French scientist in those days, French scientist Lauren King in 1878 observed the blood of a malarial patient, observed the blood of a malarial patient under the microscope, and he found and he found and he found what? He found some microorganisms in the blood of the patient, in the blood of the patient. He was also, for this achievement, he was awarded the Nobel Prize for discovering these microbes that causes the malaria. Later on, about 5 years later, those microbes were given the name as, those microorganisms were given the name as Plasmodium. What was given the name? As Plasmodium. Now students, with this all, the one phase in a biological problem, this was ended. Like for example, the observation history that was about 2000 years earlier in the Greece era, and nowadays about 200 years earlier. So the microbes were discovered later on, which was given the name as a Plasmodium. So this is the end of the observation. Now students, next phase in solving a biological problems, that is the hypothesis. In our previous lesson, we have already defined what the hypothesis is. So the possible hypothesis and deduction. The hypothesis which was made about the malaria, that was from the observation, it was made that if that Plasmodium is the cause of Plasmodium is the cause of malaria. So this was the hypothesis. So next phase comes, that is the deduction. Student, as we all know, deduction, it is a testable statement based on the previous studies. So the possible deduction for the previous studies could be, if Plasmodium is the cause of malaria, if Plasmodium is the cause of malaria, then all the people suffering from malaria suffering from malaria should have plasmodium in their blood. Should have plasmodium in their blood. Next phase come in solving a biological problem is the experiment. Students, for testing the deductions, two groups were made. Like for example, if plasmodium is the cause of the malaria, then all the people suffering from malaria should have plasmodium in their blood. This deduction was made. So next phase comes the experiment. Experiment, it is the last phase in the solving the biological problem, malaria. So students, in experiment two groups were made. How many groups were made, student? Two groups were made. One was control. Group 1, they were the control group. Group 2, that was the experimental group. The control group, this was based on the healthy people. Control group was based on the healthy people, and experiment group was based, and experimental group have malarial patient. Students, so when the blood of both of the group was tested, when the blood of both the group was tested, the group was tested. All the people suffering from malaria, all the people suffering from malaria, have plasmodium in their blood. And the people of the control group are the people of the control or healthy group have no malaria in their blood. So student, it was concluded that plasmodia, it is the cause of the malaria.